Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on signal transmission at synapses. Now, today in this lecture, we are going to learn how the nerve impulse propagates through the synapses. And the learning objective of this presentation lecture that we are going to learn about uh, two types of synapses. One is electrical synapses and the second one is the chemical synapses. So in both these synapses, how the nerve impulse propagate and what are the structure of these two synapses that we are going to discuss today. Okay. Now, if you recall the topic of muscle contraction, in that we have learned that when we are contracting our skeletal muscle, that time the nerve impulse from the nerve propagate into the muscle, and that junction, that synapse, was known as the neuromuscular junction. So between the muscle and nerve there is a neuromuscular junction as a synapse. So synapse, where actually the synapse are present. So synapse is present between, generally between the two neuron, but it is not only the case that between the neuron the synapse is present always. So but the synapse can be present between the neuron and the effector cell, and the effector cell is the muscle cell or the granular cell. So synapses are present between the neurons or between the neuron and the effector cell. Now the term presynaptic neuron, pre means before. Okay. So presynaptic neuron is a neuron which is carrying nerve impulse in the direction of synapse. Okay, presynaptic neuron is propagating nerve impulse in the direction. Presynaptic neuron is sending the nerve impulse toward the direction of synapse. Okay, and presynaptic neuron is a cell that sends a signal. So the cell is sending the signal, it is sending the nerve impulse. Now the second type of neuron is a postsynaptic neuron, and postsynaptic neuron is a cell which is receiving the signal or the nerve impulse. Okay, so the postsynaptic cell can be a neuron. It can be a neuron which is receiving the nerve impulse, or it can be a effector cell, muscle cell, or the glandular cell, skeletal muscle cell, or the cardiac muscle cell, or the smooth muscle cell. It can be. Okay. So here in the figure, in this figure, as you can see, these are the two, this one and this one, these are the two post ganglionic neurons are shown, and this one is the presynaptic neuron, and this one, this is the axon terminal. Okay, and at the end of axon terminal, this one is the synaptic end bulb. So here you can see the synaptic end bulb is forming synapse with the soma of the post ganglionic neuron. So these are the synapses. This one, this one, this one. These are the synapses. And as you can see, both these neurons are unmyelinated and they are making synapse. Okay, so here we can see this is the axon terminal, this one is axon terminal, these are all axon terminal, and at the end of axon terminal there is a synaptic end bulb, and now here the synapse is made between the neuron and the glandular cell. Okay, glandular cell is acting like the effector organ cell, and here the neuron can be formed between the synapse. The synapse can be formed between the uh, nerve cell and the muscle cell. So this is the smooth muscle, and this one is the synapse, and this can be a skeletal muscle or the cardiac muscle cell, and this can be a synapse. Okay. Now in this figure, as you can see, this red in color. This cell is the post synaptic neuron, and with this cell, as you can see, numerous presynaptic neurons are forming the synapses. So this is the axon terminal. This part is the axon. These are the axon terminal. This 1 2 3, and these are the synaptic and bulb. So all this blackish dot are the synaptic end bulb, and where they are present, so they are present on the dendrite. That means the synapse is formed on the dendrite. Okay, and synapses are also formed on the soma or the cell body. Okay. Now in this figure, see, this one is the axon terminal. Okay, axon terminal. That means this cell is the presynaptic neuron, and this bulge portion, this portion. Okay, this is the synaptic end bulb. This is this portion. This bulgy portion is known as synaptic end bulb, and this cell, this is the postsynaptic neuron, and this cell is the presynaptic neuron, and this part, this portion, okay, this portion is known as the synapse. Okay, so synapse is the point where that generally the two neurons are communicating with each other, and they are involved in propagation of nerve impulse. Okay, so this is regarding the synapse. So in this figure you can have idea regarding various kind of synapses. Okay. Please read all the names. Okay, all the names are important. Now we are discussing the third point. Now here in this figure also, okay, see, many neurons are shown, and you go through this figure and you will realize that there are various synapses are present, and you see the manner in which the neurons are communicating with each other, and the way the nerve impulse propagate, that is shown by the black arrows. So the black arrows are showing the direction of the nerve impulse. Okay, so there are converging circuit is there, then the diverging circuit is there, a reverberating circuit, then parallel up after discharge circuit is there. Okay. Now the types of synapse. See, most of the synapse, they are present between neurons. Okay, and when the synapses are present between the neuron, they are known as exo dendritic synapse. Okay, now exo dendritic means, see, exo dendritic means from axon to dendrite. So in this figure See, this is the axon of presynaptic neuron. Okay, this neuron is postsynaptic neuron. This yellow color are the dendrites of postsynaptic neuron. These are all axon terminal. Then these are the synaptic end bulb. Okay, and these are the these are all these are all synapse. Okay, so these synapse are known as exo dendritic synapse, which is present between the synaptic end bulb of presynaptic neuron and the dendrite of postsynaptic neuron. Then another type of synapse is exosomatic, and in this exosomatic, as you can see, uh, these are the axon terminal, synaptic end bulb, and synaptic end bulb, they are forming synapse with the soma of post synaptic neuron. So this type of synapse are known as exosomatic. And third type of synapses are exoaxonic. So in exoaxonic, synapses are formed between axon and axon. So see, this is the axon of presynaptic neuron. These are all axon terminal. Then these are the synaptic end bulb. This is axon of postsynaptic neuron, and these are the synapses. Okay, these are the synapses. So this synapse between axon, they are known as exoaxonic synapse. So three type of synapse: exodendritic, dendritic, exosomatic, and exoaxonic. Okay. Now we are discussing about the electrical synapses. Okay, what is electric synapse? See, electric synapse, where this type of synapse are present. So electric synapse occur between cells that are electrically coupled via gap junction. Okay, so you will find electric synapse in between those cells in which the nerve impulse propagate in the form of electricity or the current. Okay, now in this type of synapse, in electrical synapse, the exolemas, that means the plasma membrane of the axon, so exolema of two neuron, they are nearly touching each other. But when we say nearly touching, that means there is some distance is present between these two exolemas, and this distance is 3.5 nanometer. Okay, and in this exolema of two neuron, they are present gap junctions. And what are these gap junction? What is the structure of gap junction? So the gap junctions are actually the ion channel, and this ion channel are aligned one upon the other. Okay, so this ion channel, they are precisely aligned channel, and they are forming the pores. Okay, and from the pores, the ions and other small substances, they can travel from one cell to another cell. Okay, so due to this presence of gap junction, the electrical current flow directly from exoplasm of one neuron to that of the next neuron. So here, see, let's say this is the presynaptic neuron, this postsynaptic neuron, this one is the exolemma, okay, of presynaptic, this is the exolemma of post synaptic neuron, and between this two exolemma here you can see these are all gap junction. Now let's see the structure of gap junction. So in the structure of gap junction, you can see this is the ion channel, and the ion channel, they are aligned one on the other. Okay, and then here the ions, so ions are shown with the purple dots. Okay, so in electrical synapse, the ion and other substance, they can travel in both the direction. Okay, so ion can enter over here, or from here the ion can move to this cell. Okay, similarly for other substances we can think of the same way. So other substances are also traveling in both the direction through the gap junction. So I am showing you the entire figure so that you can have idea of the electrical synapse. So on left hand side they are showing us the electrical synapse, and on right hand side they are showing us the chemical synapse. Okay. Now the this arrangement of gap junction between the two neuron creates two unique feature of electrical synapse. See, synaptic transmission is bidirectional in electrical synapses. In electrical synapse, transmission is the nerve impulse propagate usually in two directions. So it is generally bidirectional. That means either neuron may act presynaptic or postsynaptic neuron, and that the current may flow in either direction between the two cells. So let's say, uh, see, if this cell is acting like a presynaptic neuron, so that time the electricity or the current will move in this direction from this cell to this cell through the gap junction. Okay, and in case this cell is acting as a presynaptic neuron, so electricity or the current will flow from this cell to this cell through the gap junction. Okay, so that's why the term bidirectional is used for electrical synapses. Okay, because the current or the nerve impulse can propagate in both the direction. Okay, now synaptic transmission is nearly instantaneous. That means the propagation of nerve impulse, that the traveling speed of nerve impulse is very fast. See, when the nerve impulse propagate, presynaptic neuron first depolarized. Okay, then the wave of depolarization finally reaches to the synaptic end bulb of presynaptic neuron, and then some chemical changes in the synaptic cleft, and due to this chemical changes as the neurotransmitter are released, so now the potential of the postsynaptic neuron changes. Okay, so in all this, see see in all this activity, for depolarization of presynaptic neuron and the potential change of postsynaptic neuron, in this process the time taken is very less, so only zero point 0.1 millisecond time is taken for this depolarization of presynaptic neuron, and after that the potential change of the postsynaptic neuron. Okay, so you can understand this is a very quick movement. So the nerve impulse, we can say, is propagating very speedily, it is moving very speedily, it is moving very fast. So nerve impulse is extraordinarily fast in electrical synapses. And we will see that the transmission at most chemical synapse require from 1 to few millisecond of time. Okay, so we are also going to compare the speed of propagation in propagation the speed of propagation of nerve impulse in case of electrical synapse with the chemical synapses. Now, the feature of electrical synapses. Electrical synapses allow the activity of group of cell to be synchronized. When stimulated, the cell will produce action potential in unison. Now the meaning of this point is that many neurons, they are connected by electrical synapse, and all this neuron, they are propagating nerve impulse very speedily, very fast. Okay, so the neuron in which there are the presence of electrical synapses, they generally work together, and they propagate nerve impulse very fast. Now, where these electrical synapses are present, so electrical synapses are found primarily in the areas of the brain that are responsible for program automatic behavior such as breathing. Now, breathing, when we say breathing, that means inhalation exhalation process, breathe in breathe out, and in this breath in breath out, we know the muscles are involved. Okay, so this muscle, they are innervated by the electrical synapses. Okay, then these electrical synapses, they are also present in the developing nervous tissue in the embryo and fetus. So during 9-month mother's pregnancy, when the baby is developing in her womb, that time in the developing nervous tissue, large number of electrical synapses are present. Okay, and during the pregnancy period, in the developing nervous tissue of embryo and fetus, it is thought that this assists in the development of the brain. Okay, so the more number of electric synapses are present, so it will be helpful for the development of the brain. Now, in addition, the electrical synapses found outside the nervous system, so outside the nervous system, they are present in the cardiac and visceral smooth muscle. So in our heart, we know there are the total 4 chamber, 2 atria, 2 ventricle. So in the wall of atria and ventricle, these electrical synapses are present. And in the visceral smooth muscle, visceral means the bodily organ, so the body organs muscle, smooth muscle, let's say in the wall of esophagus, then in the wall of stomach, small intestine, large intestine. So in all this GI tract organ, in all this GI tract smooth muscle, the electrical synapses are present. Okay, and as they are present in all these tissues and organ, they allow these tissues to engage in coordinate muscle activity. Now when we say coordinated muscle activity, though in case of heart we can say it is the beating of heart, and when we say the coordinated muscle activity for visceral smooth muscle, that means the process of swallowing, then the peristalsis process. So in that process also these electrical synapses are involved, they are playing role, they are propagating the nerve impulse. Okay. So in short, when we say electrical synapse, that means between two neurons, the gap junction is present. What is gap junction? It is the ion channel aligned one upon another, and from this ion channels or the gap junction, there is the movement of ion in both direction, and same way the electric current can move or pass in both the direction. Okay, and any of the two neuron among the any of the anyone among these two neuron can act as presynaptic or the postsynaptic neuron. Okay. Now we are moving forward. So with this we have completed the discussion regarding the electrical synapse. Now this is the figure on left hand side. In this figure you can see the electrical synapse, and on right hand side the chemical synapse is shown. So now we are discussing chemical synapses. Now in chemical synapse, plasma membrane of presynaptic and postsynaptic neuron are very close, but the plasma membrane of presynaptic and postsynaptic neuron, they do not touch. That means there is some distance between these two plasma membrane in chemical synapse. Okay, so these two plasma membrane, they are separated by a region known as synaptic cleft. And what is synaptic cleft? Synaptic cleft is a space of 20 to 50 nanometer, and in this space interstitial fluid is present. Okay, so here in this figure, see, this is the axon of presynaptic neuron, this one is the axon of postsynaptic neuron, and here they are showing this one is the axon terminal, this is the synaptic end bulb, and this one is the dendrite of postsynaptic neuron. So synaptic end bulb, and between the dendrite there is the presence of chemical synapse. So this is the axon terminal, this one is the synaptic end bulb, this is the axolemma or the plasma membrane. Okay, this one of presynaptic, and this is the plasma membrane or axolemma of postsynaptic neuron. Okay, and this distance between this presynaptic and postsynaptic neuron, this distance is known as the synaptic cleft. Okay, so here the synaptic cleft is shown, and what is there in synaptic cleft? So interstitial fluid is present in this synaptic cleft. Now the nerve impulse cannot conduct across the synaptic cleft. Nerve impulse cannot cross synaptic cleft. So to propagate this nerve impulse in postsynaptic neuron, an alternative indirect communication is required. Okay, so here you can see the action potential or the nerve impulse has reached to the terminal part of the axon, and the nerve impulse has entered into the synaptic end bulb. But now the problem is this nerve impulse cannot directly travel into the postsynaptic neuron. This nerve impulse cannot cross the synaptic cleft. So here now there is a requirement of an alternative method by which the nerve impulse propagate to the postsynaptic neuron. And now what is this alternative method that we are now discussing? Now you first you observe this figure. Okay, this figure is showing events at a chemical synapse. So with the 1 2 3 4 point, they have shown the events that are taking place at the chemical synapse. Now in response to nerve impulse, when the nerve impulse travel through the presynaptic neuron, that time and when the nerve impulse reaches to the end of the axon terminal, or the when the nerve impulse reach to the synaptic end bulb, that time presynaptic neuron releases a neurotransmitter. Now this neurotransmitter which are released by presynaptic neuron, this neurotransmitter diffuses through the fluid, interstitial fluid which is present in synaptic cleft, and this neurotransmitter then binds to the receptor. And where this receptor are present, so they are present in the plasma membrane of postsynaptic neuron. Okay, so in the form of neurotransmitter, now the postsynaptic neuron receiving the chemical signal, and now in turn they produce a postsynaptic potential. So as the neurotransmitter is combining with the receptor on plasma membrane of postsynaptic neuron, so there is a potential change in the postsynaptic neuron plasma membrane, and this potential change is known as postsynaptic potential, and it is a type of graded potential. Okay, so now we can say the presynaptic neuron is converting an electrical signal, that means the nerve impulse is converted by presynaptic neuron into chemical signal, and this chemical signal is actually a release of neurotransmitter, and this neurotransmitter or the chemical signal is received by postsynaptic neuron, and this postsynaptic neuron now generating the electrical signal, it is generating the nerve impulse. Okay, and that is known as postsynaptic potential. Now now the time required for all this activity, all this process at chemical synapse, so the time requires is about zero point 0.5 millisecond. So now you can realize that when we were discussing the electrical synapse, and in electrical synapse nerve impulse propagate in the form of electricity or the current, so that time the speed was only 0.1 millisecond. But here in case of chemical synapse, the chemical or the neurotransmitter is involved, and then the potential change is taking place in the postsynaptic neuron. So for all this some delay occur. Okay, and this delay is taking the time of 0.5 millisecond. Okay, so the propagation of nerve impulse will take a time of 0.5 millisecond in case of chemical synapse. Okay, so so simply we can say electrical synapse they propagate nerve impulse very fast, and the chemical synapse they propagate nerve impulse slowly. Okay, now now we are discussing the events that are taking place in chemical synapse. Okay, so now one by one I'm showing you. See what is happening. First you see this two cell. Okay, I'm showing you this entire figure, and this figure is showing all the events at chemical synapse. So these are the two cell. Okay, one cell, this cell, this cell is the presynaptic neuron, this cell is the postsynaptic neuron, and these are the axon terminal, these are the, and then these are the synaptic end bulb, these are all, and these are the dendrites of postsynaptic neuron. So between this synaptic end bulb and the dendrite there is chemical synapse. Okay, and you see the direction of nerve impulse, so from presynaptic neuron to postsynaptic neuron. Now you observe this figure. See all the events. Okay, now one by one uh, we are trying to understand this entire mechanism. See, first what is happening, nerve impulse reach at the level of axon terminal, nerve impulse enter into the synaptic end bulb. So this bulgy portion, this entire bulgy part, okay, is known as synaptic end bulb. So nerve impulse has reached to the level of synaptic end bulb. Now here 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 the fluid is present, and this fluid is known as extracellular fluid, and in this extracellular fluid Ca twice plus ions are present in some good concentration. So the concentration of calcium ion are very high in extracellular fluid. Now in the exolemma you can see voltage gated calcium channels are present. Okay, so when the nerve impulse reach to the synaptic end bulb, that time this voltage gated calcium channel open, and now the calcium ion from extracellular fluid, they enter, calcium ion they enter into the synaptic end bulb. Okay, so calcium ion are entering into the synaptic end bulb. Now what is happening, as the calcium ion enter into the synaptic end bulb. So here in the synaptic end bulb you can see the synaptic vesicle are shown. These are the synaptic vesicle are shown, and in this synaptic vesicle they are filled with thousands of molecule of neurotransmitter. Okay, now the exocytosis of this synaptic vesicle occur. So this synaptic vesicle they are reaching to the end. Okay, they all are aligned over here, and now this synaptic vesicle they are bursting. Now they, when they burst, so the neurotransmitter is released. Okay, so here with the red color you can see the neurotransmitter is released, and the neurotransmitter is acetylcholine. Okay, now this neurotransmitter, where it is present, so right now it is in the interstitial fluid, it is in the interstitial fluid which is present in synaptic cleft. Now this neurotransmitter they diffuse through this interstitial fluid, and then they reach to the opposite direction. Okay, so in opposite direction, this we can say is the exolemma of postsynaptic neuron, and as you can see in this plasma membrane the ligand gated channels are present. Okay, these are all ligand gated channels. Now on this ligand gated channel receptors are present. So these are the receptor. As you can see, small pit you can see. So this small pits, they are acting as neurotransmitter receptor. Okay, and this neurotransmitter joins with this receptor. So once the neurotransmitter acetylcholine joins this receptor, what happens? This ligand gated channel, they are now open. Okay, and as the channels are open, what happens? The Na plus which is present outside, this Na plus ion, they cross this ligand gated channel and they rush into the cytoplasm of the postsynaptic neuron. Okay, so see, Na plus are entering. Okay, now one more figure I am showing you. So here also you can see acetylcholine. Okay, acetylcholine neurotransmitter bind with the receptor, so ligand gated channel open. Okay, and the Na plus is entering into the postsynaptic neuron. Here you can see Na plus is entering to this channel. Okay, and the entry of Na plus is very fast, it is very rapid. Okay, so many Na plus, they enter very quickly in the postsynaptic neuron. Now as the Na plus enters, and where they are accumulated, so they are accumulated inside the inner side of the plasma membrane of postsynaptic neuron. So they are accumulated over here, and Na plus, they are positively charged. So what happened? So on outside plasma membrane, the negative charge develops. So in this portion we can say there is a depolarization occur in this plasma membrane. Okay, so plasma membrane is depolarized, and now for repolarization, the K plus ion, okay, for repolarization, the K plus ions are moving out from the channel. Okay, so this K plus, they are also traveling through the ligand gated channel, and here we are not misunderstanding that these are the two separate channel. These are the same. This one is the same channel, but for simplicity to make us understand, that's why they have shown separately, but actually it is the same channel. And in this channel, Na plus enters into the postsynaptic, and the K plus, they are moving out. Okay, and but the influx of Na plus is very rapid when we compare the outflux of K plus. Okay, K plus are moving out slowly, so that's why here the depolarization occurs. Okay, and when the K plus, they are moving out, that time this will be again, there will, this outside plasma membrane will be once again positively charged, and once again here there will be negative charge development. But before that, now this portion is going to depolarize. Okay, then this portion is going to depolarize, then this portion. Okay, so this wave of depolarization repolarization is reaching toward the axon terminal of postsynaptic neuron. This we can say the potential of postsynaptic neuron is changing. Okay, due to this depolarization repolarization, and the nerve impulse is propagating, and it will trigger the axon terminal of postsynaptic neuron. Okay, now once the nerve impulse propagate through the postsynaptic neuron, now the role of ACH, acetylcholine neurotransmitter, it role is over. Okay, now what happens to this acetylcholine? So in the synaptic cleft, enzyme is present, and that enzyme is known as acetylcholine esterase. Okay. Now this ACH combined with this enzyme acetylcholine esterase, and this acetylcholine esterase degrade the acetylcholine. So this acetylcholine is degraded into acetate and choline, and this acetate and choline, they once again they enter into the synaptic end bulb of presynaptic neuron, and they are further utilized for reproduction of neurotransmitter acetylcholine. Okay, and it is not that all the acetylcholine combine with combined with this enzyme acetylcholine esterase. It is not the case. Okay, and it is not that all the choline and acetate they return into the synaptic and bulb. So now what happened to if what happened if in the synaptic cleft the excess ACH and the acetyl and this acetate and choline are present, what happened to them? Okay, so it is believed that this ACH and choline and acetate they move in the extra they move in the extracellular fluid, and once they move in the extracellular fluid, so now they are taken up by some special kind of cell. Okay, so this special kind of cell in the CNS, this cell are the astrocyte cell, and this astrocyte cell they absorb the stray neurotransmitter, and this astrocyte they are returning this neurotransmitter ACH into the synaptic end bulb of presynaptic neuron. Okay, and in case of PNS, so in the ganglia of PNS, it seems that the satellite cell surrounding the neuron they are performing this role. This role means they are uptaking the neurotransmitter, and then they are sending back this neurotransmitter into the presynaptic neuron. Okay, now all the theory point and uh, figure I would like to show you one by one. Okay, all the figure I am showing you. So this figure we have seen. Okay, now these are the see structural types of synapses, the three types of synapse. Okay, now here one table. In this table classification of neurotransmitter, just in case you are interested. Okay, so chemical structure, function. Now in this figure also, okay, structure of acetylcholine. Now one more table for neurotransmitter. Chemical synapse. See all the steps. Okay, one by one they have given all the steps, all the events at chemical synapse they are showing. the big picture of chemical synaptic transmission. Okay. See, these are the events 1, 2, 3, 4, 5. Okay. Then 6, event number 6, 7. Okay. See. Okay. Synaptic relationships between neurons and the direction of signal transmission. Okay. In this figure, synaptic relationship between neurons. Okay. Axon terminals synapsing with the soma of neuron in marine slug. Aplysia. Scanning electron microscope, SEM. In the neurotransmitter. Okay. Chemical synapse. Structure of chemical synapse. Okay. Then the table for neurotransmitter. Selected examples. That means there are so many neurotransmitters are there, only few selected examples they are giving us. Structure of neurotransmitter. Okay. Transmission at a cholinergic synapse. See, one by one, all the events at chemical synapse 1, 2, 3, 4, 5. Okay. Then point number, see, then cessation, how the nerve impulse, how the signal sees. Okay. What events after the nerve impulse has traveled through, then what happened? So cessation of the signal, all the events. Okay. See, we have discussed this neurotransmitter degradation, reuptake, then the diffusion. Okay. So yeah, with this we have completed this presentation lecture. Okay. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.